Welcome to the online learning. In today's class, we are going to learn about the effective methodologies that are used in the preservation of milk and milk products. So, here we are going to learn about three effective methodologies such as pasteurization, sterilization, and dehydration. To give you a brief introduction about why exactly sterilization is important, especially when it comes to the matter of milk, we know that milk is believed to be highly nutritive in nature, which is also believed to be a perfect habitat for the organisms to grow. Irrespective to their pathogenicity or its normal microflora, it is important to sterilize them to keep them away from any sort of microbial contamination. When we take the milk, it is believed to be the lactal secretion of the cow, usually, and also cow milk from the goat, milk from the buffaloes are used for manufacturing of various milk products. Here, milk is available in various forms based on their fat concentration. Based on their percentage of fat content, we categorize milk into various types such as homogenized milk, reduced fat milk, low fat milk, fat free milk, that is the skimmed milk. To speak about the temperature that is involved in the pasteurization or a simple sterilization process is about 63 degrees Celsius to 105 degrees Celsius. We know that as we increase the temperature, the time duration that is required reduces. So they are inversely proportional. So one such effective methodology in sterilizing the milk is pasteurization, which was invented by a French chemist called Louis Pasteur. Thereby the name comes pasteurization. This is one of the primitive methodology that was invented yet very beneficial in still today's life. When I speak about pasteurization, it is application of the heat of appropriate temperature to appropriate time. Here, this completely aims in reducing the pathogenic count and also the organisms that can cause spoilage of milk. So the pasteurization is again of various types where we call them as VAT pasteurization or the low pasteurization, where the temperature that is required to pasteurize is minimal, that is 63 degrees Celsius, and the time duration is about 30 minutes. And next we have high temperature and the short time, where we require about 72 degrees Celsius for 15 seconds. Ultra high temperature, that is the ultra high pasteurization, we use the temperature about 138 to 150 degrees Celsius, that is for 1 or 2 seconds, and ultra high temperature, that is about 280 degree Fahrenheit for 2 seconds. We always tend to assume that pasteurization is meant for milk. Apart from milk, we also pasteurize various other products such as beer, like canned goods, dairy products, egg, juice, nuts, syrups, vinegar, water, and even wine. The main methodology, the objective behind this methodology is to destroy the pathogenic organisms that are present in the milk by the process of milking or packaging or processing, and also to ensure the quality of the milk is to its fullest, also to destroy the unwanted microorganisms present. When we speak about the types of pasteurization, high temperature, as we mentioned, they are of varying temperature with time. When I say ultra high temperature or ultra pasteurization, we have about 280 degrees Celsius, which sometimes not only harms the organism but also coagulates the proteins. They also damages the vitamins, minerals, and also some sort of protein globules that are there in the milk. High temperature pasteurization kills the enzymes, inactivates the enzymes, but also gets rid of all the pathogens that are there. And low temperature pasteurization leaves the proteins or the enzymes untouched, whereby removing only the microorganisms. 108 degrees Celsius is the temperature where is most friendly, but above which the protein starts denaturing. So here these are the four types of pasteurization based on their temperature and the time, which is inversely proportional. So the advantages is maintains the quality of the milk, makes the milk free from any sort of pathogen. And also, what are the disadvantages would be it diminishes the cream line or the cream volume. Here pasteurized milk may also vary the renetting time, and also it fails to touch the bacterial toxins. At times the bacteria is not the one which causes the disease, but it toxins. But here during pasteurization the toxins are left untouched, and also because of the high temperature there is a denaturation in the protein, alteration in the enzyme or in the protein globules make them less nutritive in nature. So using an appropriate temperature is very, very important. And the next methodology that we're going to see in today's class is going to be sterilization or ultra high temperature, which is mainly focusing on pasteurizing the milk, as in sterilizing the milk in such a way that they can be saved or stored in the room temperature henceforth. After the pasteurization or sterilization process, after this sterilization process, it is now fit to be stored under the room temperature. This ensures that destruction of complete microorganism includes pathogenic organisms and also the organism which causes spoilage. Apart from that, it also removes the vegetative viral particle and also the viral form spores or fungal spores and the vegetative spores. To speak about the methodologies that are involved in sterilization, they can be the conventional method where they're gonna bottle the milk and then pasteurize them together of about 105 to 110 degrees Celsius for 30 to 45 minutes. At times, they are going to be sterilized in a big rotator or a drum for about 135 degrees Celsius to 150 degrees Celsius for about 1 to 2 seconds, and then fill them in the sterile bottles. So that is called as the aseptic methodology. In bottle sterilization or the conventional methodology is when you pack them and then sterilize them. So these are the simple methodology that are involved in your conventional method and the aseptic method. To speak about the sterilization in mass, here we are going to sterilize the milk in the mass concentration, quantity in the horizontal retort or your vertical retort. And then we have the heat treatment methodology, which is believed to be one of the important methodology where dehydration of the milk occurs, thus obtaining the dried milk or the powdered milk or the condensed milk. And but here we are going to mainly focus on the powdered milk. The main objective is to ensure complete removal of water, thereby you're removing the organisms, because the minute you remove the water quantity, it is becomes a improper or unfit habitat for the microorganisms to reside. So thereby increasing the shelf life of the milk, and also reduce the volume or the weight of the milk, where the nutritive nature in the milk can be concentrated. This happens via three ways, where we have drum dryer, spray dryer, or fluid bed dryer. Where in the spray dryer we are going to pre-concentrate the liquid food, that is the milk, and disperse it through the hot gas. Thereby, because of the evaporation in the water due to the high temperature, there is a concentration in the milk quantity. Drum dryer, we have two rotating drums which rotate around, which is thus connected to a heater. Because of the internal heating coils, the drum concentration, the temperature would increase, thereby removing the water. Fluid bed drying methodology involves a cooling, agglomeration, granulation, and coating the particle material to its air dried form. Thus, the globules are also protected. The milk is now thus powdered, which is the dehydrated milk. This is a simple methodology that is maintained in the industries to obtain the powdered milk, especially for the neonates. The first thing is the collection, quality analysis, that is the separation and standardization based on their quality, and also the quantity of the solid not fat content and the fat content, and also about the normal proteins present in them. Pasteurization process, of course, before we go ahead with the production of powders, we heat them, we sterilize them, as in we pasteurize them, and then we preheat it in such a way that the evaporation process starts. And in the ultimate evaporation process, which happens around 72 degrees Celsius, the water is completely removed from the milk. We know water is the most a major constituent of the milk. When that is removed, all the other mineral constituents are left behind. And then the homogenization is not really done usually, but it is also done in order to obtain the whole milk or the buttermilk being processed properly to decrease the fat content. By heating, here we have the drying chamber, which ensures the leftover water to be evaporated, increases the energy available for drying, and also it decreases the viscosity of the thus dried milk. And also air filtering prevents the contamination process, or any sort of dust or any particular matter can be removed. And then we have the feed pump. Three piston pump with a particular pressure is used to pump the feed. So this atomization is a very important process, where this atomization is a process where we are going to produce droplets of milk powder, that is about 10 to 20 200 um, where about 40 to at least 80 should be produced, where this water droplets results in the higher concentration area, which aids in drying. The minute they are atomized, they are not easily dispersed off. They will sit retained inside the holder, so this atomization would keep the powder intact. So the loss of milk powder or the concentrated form wouldn't be wasted. And then we are going to introduce them to the drying chamber, where we introduce them to a hot air that is blown, and then they ensures complete removal of water. Then cyclone separator is based on the centrifugal force exerted on the particle. This cyclone separator is very important in the maintaining the density of the particle, finds or sends forward to the filter henceforth. Here we know water is the main constituent of the milk after it has been brought down into a powdered form. Except the water, everything else remains constant, where it is also in a much more concentrated way. Where we were speaking about the drum drying, this is the way which is most effectively used. We have two drums that is rotating around, which is fitted with the internal heating coils. Because of the increase in the temperature, they are going to evaporate the water content in the milk. Thus the concentrates of the milk resides on the top of the drum. They are being skimmed off using the or scraped off using the knife blade and stored in the storing tank. So this is an important methodology over the fluid bed or other methodology. This is most commonly used in the industries, most effective, easy methodology, and less time consuming comparatively. Thus packaging and storing becomes an again important part, where the milk is either packed in the plastic or a multi-walled bag, or an airtight bag, which is also filled with the nitrogen gas, which acts as a preservative gas, which will not allow the oxidation of the contents in the milk, and also maintains the flavor of the milk, thereby increasing the shelf life of the milk, maintaining its quality as well. Packaging is always a challenge, because you should keep it away from any sort of contaminants, moisture, sunlight, air, oxygen. So packaging them and storing them in a right way is very required. The bags in which we store the thus obtained powdered milk has to be of several layers so that they provide a mechanical barrier to come in contact with any sort of unfavorable condition or the contaminants. So packaging and storing and transportation is a thing that has to be equally concentrated about. So these are the three important methodologies that we have learned in today's class, which includes pasteurization, sterilization, and dehydration, where the dehydration directly correlates to the dried powdered milk. So these are the three important effectively used methodology in the industries to process milk and preserve milk. That is all we have for today's class, and thank you.